you so much, Doctors Rasheed, Wang, and Patel. So for our audience, if you guys would like to ask a question and hopefully get it answered today, we're going to answer as many questions as we have time for. Go ahead and put your question into the Q&A chat pod, and we will go ahead with that. We'll go ahead and get started. So our first question is for Dr. Rasheed. You mentioned that demonstration of accuracy using a low molecular weight cutoff membrane for recovery of drug has practical challenges due to low concentration and drug absorption. Could you please clarify what the expectation is for this value?
Thanks, Lisa, and thanks for the question. It's a very good question and very pertinent one. So, as it was discussed during the presentation, that determining drug in aqueous phase using low molecular weight membrane is challenging. And of course, it's challenging for two reasons. A, that it is anticipated that drug is going to be at a very low level in the aqueous phase. And secondly, the choice of the membrane filter could contribute due to adsorption. However, we do expect that you do validation for accuracy using both high and low molecular weight membranes. So using low molecular membranes, there are two possibilities. One possibility is that you are going to detect a very low or minuscule amount of drug. And second possibility is that you don't detect anything at all. So the question is that, like, how do we have confidence in those results, whether you are detecting anything or you are not detecting anything? So that's why one suggestion that was part of the presentation, that the analytical methods that you are using to assay the drug in different phases, they also need to be adequately validated. So in case of accuracy, using low molecular weight membrane, one thing you can do is to do a study to demonstrate that the method is capable of detecting and quantifying the drug at low concentrations. And one way of accomplishing is to make aqueous solutions of known concentrations for the drug. Now, I'm saying this having realized that the drugs for ophthalmic emulsions, so far the two drugs, difluprednate and cyclosporine, they are both known to be very low soluble in aqueous medium. However, the solubility is known, it is in the literature, so you can actually create aqueous solutions of known concentration and that you can assay, and that you can use those solutions to establish LOD and LOQ of your method. And then you can pass through that solution through the low molecular weight membrane filter, and then assay the filtrate, and that will give you some idea to the extent of adsorption. And that way you have some confidence that if you are not detecting anything, that means the level of drug in the filtrate is below the detection limit. So I hope that would sort of help you at the use of the lower end of the molecular weight membrane filter. Thanks.
Great answer. And our next question is going to be for you also. Are the validation requirements discussed the same for other types of drug distribution methods, such as ultracentrifugation or dialysis?
Thanks for the question. Again, great question. So the general principles which were discussed during the presentation, they apply. So any methodology that you're going to adopt, so you need to be thinking the details and the nuances of those methods. And in terms of specificity, accuracy, and the suitability of this method for the intended use, and keeping in mind the intended use. The intended use is to compare your test product and the reference product for the amount of drug in different phases, right? So having said that, any method that you adopt, you have to pay close attention to the details and think on the considerations for validation, and like so it's like your diligence as part of method development. So I hope that answers your question. Thank you.
Thank you. Our next question will be for Dr. Wang. Can an IVRT or dissolution method developed for quality control also be used to support bioequivalence?
Thank you, Lisa. Yeah, that's a common question we have been asked, and thank you for that question. In general, the answer, in short, is yes. You may use the same method for both BE purpose as well as QC purpose. From scientific consideration, there really is no fundamental difference between developing an IVRT for QC versus for supporting bioequivalence. That being said,
Hey, Dr. Wang, I think we're having a little bit of trouble hearing you. I think the volume is really low. So if you want to go on mute, and then our AV people will help you get the audio a little bit turned up on your side. And I think with that, we're going to go to Dr. Patel, and then we'll come back to that question and move back. But our next question will be for Dr. Patel. So for IVRT sensitivity, selectivity, and specificity, you mentioned about the selection of strength, and you specifically mentioned using 150% label claim and not 200% label claim to the nominal strength. Would you please share the rationale for the same?
Certainly, it is a good question. To conduct IVRT sensitivity, specificity, and selectivity, it requires two similar formulations by altering the concentration of the drug, that is, one with the higher strength and one with the lower strength. In my opinion, the higher strength of the drug product to be used is 150% label claim and not the 200% to the nominal strength, because the IVRT method should be able to discriminate formulations with 50 to 150% variation in the strength by comparing the drug release rate to the reference strength as being inequivalent. If the IVRT method cannot differentiate a release rate as being inequivalent until it is 200% to that of the reference strength, then the IVRT method may not be suitably discriminating.
All right, great. Thank you, Dr. Patel. And we have another question for you. You discussed selection of a specific approach for demonstration of IVPT sensitivity. I would appreciate it if you could provide insights on how the specific approach should be selected in order to conduct this.
Thank you for your question. This is a broad question, and as I mentioned earlier, one-size-fits-all approach does not apply to demonstrate IVPT sensitivity. There are multiple factors which needs to be considered here. For my understanding, in general, an IVPT method development study with different dose is well suited to topical products that contain volatile components which evaporate from the formulation following dose application to the skin. And the examples are many of the gels, lotions, and creams. However, the difference in dose amount may not necessarily produce noticeable difference in drug delivery, that is drug permeation, for topical products like petrolatum-based ointments, for which the altering the dose duration could be a potential pathway to move forward.
Great, thank you, Dr. Patel. And our next question will be for Dr. Rasheed. What is the regulatory request for outsourcing a lab for Q3 characterization? Is the GMP?
So the short answer is yes. Any lab that you are outsourcing and using for Q3 characterization, as a matter of fact, any other testing or characterization should be in the GMP status. Thank you.
Okay, great. And another question for you. It is a really big challenge for the recovery of drug when there is low concentration in the aqueous phase. If it's not possible to get the recovery, what other considerable options are there? Or what other options would
Thank you. A good question. So I think it ties into the very first question that I received, that recovery from aqueous phase, or the phase that is collected using the low molecular weight membrane. And we do recognize that it is a challenging feat. But however, it goes back to your diligence on how well you perform the validation of the analytical methods. And as long as you are demonstrating that using the low molecular weight membrane, you are separating the aqueous phase and your method is capable of detecting the drug at a reasonably low level. So as long as that demonstration is there, so that means if you are not able to recover anything from the aqueous phase after passage through the membrane, there are only two means. There are only two possible explanations. A, the level of the drug in the aqueous phase is well below the detection limit of your analytical method. And secondly is that during the course of this transfer of the filtration, the membrane filter is absorbing a good chunk of the drug. So that contributes to the lack of detectability. But however, you need to sort of provide that evidence that you have an analytical method which is capable of detecting the drug at low enough levels. And as I mentioned previously, that the solubilities are available in the literature. One can create those solutions in the lab, and then you can use those solutions to establish your analytical methods, LOQ and LOD. I hope this answers your question. Thank you.
Great, and we have another question for Dr. Rasheed. Is drug distribution in different phases a one-time?
So yes, drug distribution and its comparison with the reference product in different phases is a one-time study, and of course the study is part of the product-specific guidances for the ophthalmic emulsion, and this is like one component of the Q3 characterizations. And as I elaborated during the presentation, that number of Q3 attributes, they also become the quality attributes during the life cycle of the product. But drug distribution itself, it's just a one-time study requirement. Thank you.
Thank you. We have a question now for Dr. Patel. Does the movement of drug across the semi-permeable semi-synthetic membrane represent permeation?
The short answer is no. The movement of drug across the semi-permeable or semi-synthetic membrane represent the release of API from the formulation and do not provide information about the permeation, and for which you will be needing a human skin to conduct the IVPT studies. Thank you.
Thank you, Dr. Patel. And with that, we're going to go back with Dr. Wang. So do you want to come off mute, and I'll ask you this question one as before. Can an IVRT or dissolution method developed for quality control also be used to support bio?
Sorry about the previous situation. I hope everyone can hear me now. Okay. So the short answer is yes. You may use the same method for both quality control purpose as well as for supporting bioequivalence. That being said, I think one important point I'd like to make is the IVRT for supporting BE is a one-time study. So sometimes depends on the dosage form and the purpose. If you want to show the equivalence in release kinetics to more reflect what's happening in vivo, for instance, for the long-acting drugs in particular, then you will have to likely demonstrate comparable release in the real application duration. But then for long-acting product, for QC, you may develop a separate method, which is more based on accelerated condition.
But then again, that depends on the purpose and your intention. Even for accelerated method, if you'll be able to show a good correlation between the accelerated condition versus the real time, then that may also be acceptable for supporting B as well. Thank you.
Okay, great. And we have another question for you. How many batches of the generic product and RLD product are needed for conducting IVRT to support bioequivalence?
Yeah, in general, when in vitro studies are used for supporting bioequivalence, we recommend 3 batches each of the test and reference products. The purpose for doing that is to, assess the interbatch variabilities associated with both the test and reference products. That being said, recognizing there is a subtle difference between the physical-chemical characterization study versus an IVRT study is a performance test. Then if applicant thinks by testing 1 batch of the reference, for instance, would be sufficient for establishing drug release between the test and reference, then they should provide sufficient justification. But otherwise, in general, we strongly encourage applicants to study 3 lots just to know better the design space and what are the variations associated with the reference. Thank you.
Okay, and another question for you. Is it necessary to show the complete release or at least 80% release for semi-solid formulation while developing IVRT?
Like I mentioned in the talk, the complete release is a general expectation, and then also that has implementation on the statistical method you may use for establishing similarity in release profiles. That being said, we do recognize, for certain dosage forms, it may not be practical or it's very challenging to really meet the 80% release criteria to be considered as complete release. So whenever, you have an incomplete release situation, then you should always include sufficient justification to see, why such situation is still acceptable and be sufficient to demonstrate your product shows a comparable release behavior compared to the reference. So I think the key really is on the adequacy of your justification. Thank you.
Okay, and another one for Dr. Wang. Does this time point selection depend on the dosage form or amount drug released at the end? For example, 6 time points versus 6 hours.
Yeah, thank you for that question. I think that one, again, it depends. I think both the dosage form as well as the amount of drug release would, play a role in terms of when you decide to end your IVRT study. In general, I think the time sampling points should be sufficient to capture the entire release kinetics or key phases of your dosage form. That being said, for some products, if that is designed to have a rapid release behavior, for instance, then IVRT within 30 minutes would consider be acceptable. Well, if one is designed to have sustained release, then you need to make sure, and also if the release is multiphasic, then you need to make sure your sampling time is frequent enough to capture those different phases. As for the entire duration, there are some general recommendation or chapters for certain particular dosage forms, like Dr. Patel mentioned, USP 1724 for topicals and for semi-solids. So those all depends on the situation. Again, I think if you decide to pre-select certain sampling points, then you should always make sure sufficient justification is included. Just say why those are considered as acceptable. Thank you.
Okay, and we have a question for Dr. Patel. USP 1724 provides two options for dosing procedure for vertical diffusion cells. What is the agency's recommendation on dosing technique and procedure?
It is a very good question. For USP 1724, the dosing procedure depends on the type of apparatus used to conduct the IVRT studies. The dosing procedure is different when you follow the model A versus model B and C, and I would suggest to follow the manufacturer's protocol for the dosing procedure.
Great, thank you, Dr. Patel, and thank you to all of our speakers and panelists. And with that, we're actually going to go to break, and we will resume at 2:40. Thank you all for your